Hello?
Yeah, hi.
Hi, how are you?
Yeah, I'm doing great.
Great. Okay. Welcome to the meeting. I don't know how many other people are going to show up, but we'll get started. Did you have anything you wanted to present today, Mainak? Okay. Well, if you do, let me know. So GSOC has been submitted and is officially over, so... Oh.
Hello?
Yeah.
Yes. Yeah, can you repeat the last line? I couldn't hear you.
Oh, I was wondering if you had anything you wanted to present today.
Today, I really don't have anything to present.
Okay, thanks. Yeah, so GSOC is over and in the books for another year. And so today maybe we'll talk about the next development steps are for the sort of the whole infrastructure of what we have in terms of data, open data, and machine learning and things like that. So if people are watching on YouTube, you can get a sense of what the scope of what we have now is, and maybe we'll think in the next couple months about what GSOC 2022 should look like. So this is what we did last year. We reassessed what we had and we moved forward on that. So we'll do that. We also have a couple of other features that I have on different things I found on the internet and in the literature. I have some things to go over, some outstanding work that we have. I kind of wanted to go over the Bacillaria Aneural Cognition paper, which is coming to fruition. I think it's far enough along I can share it with people. And then we'll see who else shows up if they want to present something. So why don't we get started? I'll share my screen. So, okay. It's not letting me share my screen. Okay, let me try it again. Okay, it's not letting me share my screen. So this is maybe a problem. Let me try to go out and come back in. I'll be right back.
Yeah, sure.
Okay.
Yeah.
All right, I'm back. Let me try to share my screen again. All right, there we go. So now you can see my screen. And so this is I wanted to bring a couple things up this week that I didn't have last week. So this is the DevoZoo interface. And this was the product of Ujwal Singh last year for his GSOC project. Basically took what we had in terms of the DevoZoo and he built another resource called DevoWorm AI, and then that was his project. So what is DevoZoo? DevoZoo is this thing we operate off DevoWorm GitHub IO, and it's hosted there, and it's a repository of data for different types of embryos and developmental data. So you can see that we have the DevoWorm, or the different, we have different species where we have data for mostly microscopy. Well, I think all microscopy data. A lot of it is the embryo. I think all of it's the embryo, actually. So this is for nematode C. elegans. This is the lineage tree. So, no, actually, we have, I should restate that. We have microscopy data, raw microscopy data, which is this here. So we have for the nematode, we have the multi-view SPIM data set. We have another multi-view SPIM data set. We have another data set that I don't know exactly, I can't remember what this is, but you can learn more here. And then another SPIM. And SPIM is type of microscopy where it's a high resolution type of microscopy. So we didn't use that in this year's project because these images aren't annotated necessarily. So this actually I don't think is SPIM, but that's okay. So this is, there's more at the link. So these are the microscopy images or the movies that we have that we link to. And then we have these data sets that are processed. They're CSV files. And we have the lineage tree, cell birth and death timing, differentiation tree, cell position, and gene expression. So these are all data sets that were collected from different sources. Those data are annotated and have variables associated with them. So it's like you can pop them into any analysis engine you want. You can pop them into like a Python environment or a MATLAB environment or an Excel environment even, and do some analysis on them. So this is something that we have as well. We also have data on the sea squirt, which is the Cystidian. This is an organism that goes through a number of life stages before it gets to adulthood. So we have the embryo, which is this, it has this eightfold symmetry that when it divides into, like, I think actually it's fourfold symmetry. It divides into 4 founder cells, and then those founder cells form symmetrical parts of the embryo. And then it gets up to a couple hundred cells, and then it goes through a different developmental phase. It becomes this long tube, and then it just eventually ends up being this the sessile or the stationary organism. And so it goes through a number of different stages of its development, and this is the first stage of its development. So we have early embryogenesis in this species, and this is a microscopy movie. These are CSV data, and then this is the movie. And we also have some data for fruit fly, zebrafish, mouse, spider. Even we have movies for spider. So these are not high resolution movies, but if one wanted to go into those data and pull out some interesting, try to do some sort of machine learning enhanced cell tracking or some other type of analysis, one could do that. We also have the Bacillaria data set, which is this, we've talked about this before, where we have these movies where the Bacillaria are, moving around in the way that they do, this accordion-like movement that they do. So these are movies for that. And then we have a couple of data sets or a couple of repositories of virtual organisms. So we have morphozoans, which are the agents that we use in the Morphozoic platform. And so there's a paper and a repository with C++ code in it. So if you want to run this, this is a cellular automata simulation, and you have agents that form these. They can do a lot of different types of pattern recognition, but they also form like developmental like structures. And then we have multi-cell systems that's even less developed. This needs to be updated, but this is the stuff that where I think I showed this in one of the lab meetings, where we have this network and we look at the edges of the network to determine this the structure of the cells, and we look at how they they undergo development, and we can look and see if they form these coherent modules. So these are all different types of things that we have, and I want to update this quite a bit. I haven't had a chance to do it, but that's one aspect. So that's the DevoZoo, and I want to I know that I was at a meeting recently, and they had talked about like resources that people need. They were talking about people who, there's a biology community and there's a computational community, and there was a consensus, or there was an idea brought up at the meeting that we need a resource that links the two, that's data driven, something that emphasizes model organisms. So I think this emphasizes model organisms. We just need to make it a little bit more friendly to people coming in from maybe a developmental biology background or a computational background. They want to know more about the model organisms. So this says a lot of different model organisms. The question is why do we use model organisms? Why these model organisms? And so maybe we do more development on that part, which isn't really like a GSOC project thing. It's just, I think, within the community, we need to think about providing that to people and seeing if people embrace it. I think they will, because I think there's a need for it. Then there's this DevoWorm AI site. This is the DevoWorm AI site where you have this open source initiative. This is the thing from OpenWorm. So this is just a different color because it's DevoWorm. So this is the OpenWorm mantra here. The open source initiative to create the first virtual organism. Come join us. And then this is our, this tells a little bit about C. elegans. This is the online tool for C. elegans. And so there's a link here. Yeah, right here. I think this is for the, okay, yeah, this is, and then this one is the digital Bacillaria. So this has, I think there's some links in here to some of the stuff that's been going on with that. There's this general segmentation model. The DevoZoo method, so this is the DevoZoo part. DevoWorm Academy. So we actually had a DevoLearn, something called DevoWorm ML, almost 2 years ago now. And in fact, I think it started on this day 2 years ago. So, but we need to update that. I haven't done it in a while because I haven't had time. But I was updating it earlier this year, and it's something I think we should put together a little bit more completely. Last time I ran it, I had it week by week, and I just kind of used this time to do the course. And I tried to get a couple people from outside our usual group, so they came in and it was just on a series of topics that were maybe of interest to biologists and to people interested in machine learning. So we had people, not just myself, but other people gave lectures as well. So that's the link. That's what this is referencing. And then this history of DevoWorm, which is this where we kind of go through this group here and its history from 2014 onward. And then the numbers. So we have this. This is actually, I think, more along the lines of OpenWorm, but we have actually some latest paper blog post articles here. I don't know why the links aren't in here, but and then this is the founder, and then people in the team. I don't know how this works. Okay, and then contact us. So there's a contact form on this. So this is kind of parallel to the DevoWorm website, but this is something that we worked out for a GSOC project. So I don't have much else to say about it except that there's so we have these two things that were done last summer. Now the question is how do we integrate what we've done this year and maybe what we've done in past years more effectively? And so we have the DevoLearn platform on GitHub, and that includes the DevoLearn software, some of the other things. And so I'm just trying to think of how to integrate this more tightly and make it look a little bit better, so people can come in and start using these tools, and then maybe using DevoZoo and other resources. So if people have suggestions about that, let me know. We'll be working on it over the course of this year. And then next year, I'm not sure what our plan is. If it's going to be if we're going to focus on doing more with DevoLearn in terms of developing it, or try to develop something else. I don't want to, we could go in a new direction, more emphasizing some of the other projects that didn't get selected this year, such as the the thing on the Bacillaria project. We could do that, or I think people are working on the axolotl image or the axolotl atlas. So we'll see where that goes. But yeah, do you have thoughts about that, Mainak?
Yeah, I think Akshay was the one who was giving a presentation last week, right? About the axolotl image.
Yeah, he was showing his notebook. So he has he's working out the problem. He's getting it into a notebook where he has the sphere that he can rotate. He's just trying to put the images on it.
Yes. And from what I figured, I think Akshay would be able to map the images on top of the sphere, like a texture on top of the sphere. I guess that's what he's aiming for.
Yeah.
So maybe we'll get an update soon from him. Let's hope for the best.
Yeah. Yeah, hopefully. Yeah, so that should maybe we can move more on that in that direction, and then if we had that, we would have a new dimension to a lot of our models. So we have a lot of models that are C. elegans oriented, and then we have the Bacillaria, digital Bacillaria thing. That was actually something that Ojwal worked on as well. So that was his project in 2020 was kind of a hodgepodge, but it came out pretty well. So this is where you're tracking these small organisms, and then they're even smaller than C. elegans. And then, of course, then you would have the sphere. You would have the axolotl embryo atlas, I guess you could call it, and that would be a third thing that we'd have. So it would be nice to have that collection of things.
That would be nice.
So, yeah, I don't want to and again, it's things that we can update. I think we need to update some of these web resources, but yeah. So
yeah, a few links need to be updated, right?
Yeah, yeah,
Yes.
and yeah. So and then we also have other model organisms. So I mean, the data sets are always, there are always new data sets coming out, and there's stuff that's available that's annotated. So that's something that people might want to. I just want to make it so that people can come to the community, find things that might be, might encourage them to do some good work, either learning or research oriented, and then they can pick it up and move with it pretty quickly. And then I'm really interested to see what we can do with some of the software, because we've been working on software the last couple of years, and I don't know. Maybe we can put something together some sort of analysis and make it into a paper of some data. A lot of the models that we have been worked out for C. elegans, but that doesn't mean that we can't do other organisms as well.
Yes.
Yeah. It's great. So that's all I had to say about that. Right now, I just wanted to give a little overview of where we are. So now we need to figure out where we're going with that. So the next thing I want to talk about is, let's see. Oh yeah, I wanted to talk about this. I did a presentation this week. It was so this is the presentation here. If we go to our YouTube channel, it's right here. It's Game Theory of Developmental Processes. This was for Dynamics Days XL, which was hosted in Nice, France, and they had it online as well. So no need to go to France. I was able to do the session. It was kind of very, very early for me, but this is the recording. This is a longer version of the talk. This is 30 minutes. So if you go through it, it's, it really kind of gives you some detail of what's going on there. And it was, I think I presented this a couple weeks ago. It's where we had the, the different models of game theory, starting with this idea of an ontogenetic agent or a developmental agent, making the point that this is different from the kind of game theory you'd find in economics, and then getting into the different types of games you can use this type of approach on. So we have some things that we published. We have some other things that I kind of cases that I've made up for the talk, And ultimately, I'd like to put out a preprint on this, but we're not there yet. I have it like in a not a very good draft state. So we can talk more about that in coming weeks, but I don't have anything much else to show except to point this out on the YouTube channel. And then there's this other, so I'm going to actually go to this for a minute. So this was the session that it was part of, Games Development and Pattern Formation. And he had a number of people in this group and different topics. So this one, they talked about the long-time behavior of a PDE replicator. So PDE is partial differential equation. So this is a PDE replicator equation. So this is something they use to look at reproduction or a model of reproduction for different types of applications. A replicator is like basically the thing that replicates like genes or replicates a phenotype. And they have this, it's a theoretical concept of a replicator. And they can actually model replicators and using differential equations. In this PD replicator equation, they looked at the long-time behavior for multi-level selection in group-structured populations. So this is like a problem of like ecology and development, or of evolution, where you have structured populations and you have selection happening at multiple levels. They also talked about coevolution of cooperation and synchronization. So this is where we're looking at migration, and they're looking at these different aspects of it. They also had this talk on generalized Hamiltonian dynamics and chaos. So this is on evolutionary games, in evolutionary games on networks. So evolutionary games are games where you have evolutionary dynamics. You look at the game over time, and you try to find equilibria. And this is very much like physics-based complex systems. So there's a lot of, it's very physics-oriented. Then there's this talk, mechanochemical pattern formation in cells. So this is has to do ties very closely to the reaction to fusion stuff that we talk about a lot in this group. The Turing morphogenesis stuff. And then there was this talk, structure versus dynamics, controlling chemical communication in arrays of diffusively coupled microoscillators via compartmentalization properties. So this is where they're looking at this type of chemical system, where they're looking at like they're treating the chemical compounds as microoscillators over time. So when they say dynamic states, they mean like, dynamic behavior, dynamical systems. That's the sort of the thrust of this conference. So this was a pretty good session, and then I was in there with them. So this is it's a nice little session. I don't have much more to report about it, but we'll come back to the papers in a little bit. Next thing I wanted to show is this thing. This is something I found in a talk I went to on virtual talk I went to on Friday, which is this. It's a visualization system that, I don't know, it's some sort of company out of the Netherlands, and they're right here. They're simulating things that are happening during the transcription of DNA, and then also things happening in the cell. So these are things you would really need to have a high-powered microscope to find. This is actually an ant tunnel in an ant colony. So they're visualizing things at different scales. Here again is the, this is some sort of binding, molecular binding, and then this is like cells being innervated by motor neurons. So this is like, it's very high quality. These are animations that people have made, this group, and they're using this type of 3D modeling where they're making these movies. These aren't really based, some of them are based on microscopy images, and some of them are just animations of processes. So I think some of these are really nice. Oh, this is not a biological image, this is a lab, but you can see like what they're doing here is they're building these three-dimensional models, and they're kind of maybe showing a little bit more than the microscopy. They're showing like the process, and it's pretty flashy. So what I'm thinking about is like, I know this has been a long-standing interest of the group, is like building something similar to this for some of the things we're talking about in development, and kind of using microscopy as a seed for that. So like we have the microscopy data, we have like, we can build like a skeleton of something, and then also, not just the skeleton, but putting that in a larger context. So, we can put the data in a larger context in one of these 3D worlds. In my other group, I'm talking about, oh, hi Akshay, how are you?
Hello.
Hello. So in my other group, we've been talking pretty seriously about getting into having a VR infrastructure. And so VR and AR and XR. So VR is virtual reality, AR is augmented reality, and XR is what they call extended reality, which usually covers both of them. And so, there are different platforms for doing this, different ways to develop. And that might be something, too, that we might do through GSOC, is to get people to do development on some of these sorts of animations. We're taking some of the microscopy data and turning it into this living thing that, and I think the Axolotl Atlas would be like a intermediate step there, or we'd have like this thing you could explore, and then, you can do that, and then, at some point we would build this sort of these worlds, these developmental worlds, where you could explore them with, a headset. There's the Oculus headset where you can actually explore things that you build in a virtual world. You can develop on in a standard language, a standard platform, and you can build things and view them. Yeah, Unity framework, that's definitely compatible with that. And so, use a C. It's not something we've talked about too much in this group, but it would be a nice sort of direction to go as well. So, Akshay, I wonder if you had any updates. Did you want to update us on anything?
Oh, this week, no. I was actually busy with some college work, and yeah, I just got so much of work to do. I couldn't actually progress. Well, I actually tried with Surface Color. Maybe I would move from Plotly and use some other libraries, or even try to make a 3D visualization myself, just not by sketching it like in a graph, or just by showing it using OpenGL or something. I would try to look at other alternatives. If the Surface Color is not working, I would give it 2 more days. But yeah, next week I guess I would be having some updates regarding that. So
that's great. Yeah. Try different things. I mean, it doesn't really matter what the underlying technology is. Sometimes you just have to get it to work. Yeah, that would be great. So that's what I had to say about that. Let me go to the next thing here, which is I think we'll review the major task board here. I just wanted to go over this. I'm not going to review it point by point, but I wanted to see where we are with this. So we have some things on here finished, in progress. I probably didn't move all the things over to finished that are finished, but, So there's some outstanding things here. This Bacillaria Aneural Cognition paper, which is quite late, but I'm going to talk about that later in the meeting. This is get data from Susan's ball microscope. I think that's actually like a continual thing here that I use the hold category for things that are continual or that have parts that we have to hold on. So I'm gonna say that we have the microscopy data here, but we could get more. And then, so that's that. This fish ladder toy model of life in a lipid world. This is something that I think Krishna was working on. This axolotl embryo animations and segmentation on something that Akshay is working on, just told us about. This analysis of shapes of polygonal archaea and shaped droplets. This is something I think that Mainak is working on. And don't worry about the time on this, just, this is just like in progress. it's open. We can keep working on it as long as we want. Recruit people as DevoLearn contributors. That's something that is probably, well, in October, of course, there's Hacktoberfest. And so this is a thing that GitHub runs to get people to work on different repos. So we might be doing, last year we did Hacktoberfest for DevoLearn. We might do this again, advertise this to people, and it's a good way for people to get involved in open source. So
are there some, sorry to interrupt, are there some known bugs in like DevoLearn right now, apart from the issues which is being listed?
All right.
I think Mainak can answer that, maybe. Mainak knows.
Yeah. I think there's a message here. Oh. Mainak said the OpenGL approach sounds interesting.
I'm not sure it would be interactive.
Yeah.
Like Kotlin, but yeah, something is better than nothing, so I'll just try.
Yeah, I mean, if we want to, if we want to assign like tasks to people for our Hacktoberfest, it starts October 1st and runs to the 31st. And, we can make issues for people if we want to do that.
Yeah.
Then we advertise it. Last year I advertised it a bit, and I got people to come in, or if you know people in your classes that might want to do this, there's I think, we can advertise it there, and then, if they make I think 5 commits and get them pull requests and get them accepted as commits, then they get a t-shirt or something. So they give a little bit of incentive.
Oh yeah, from the Hacktober team, yeah?
Yeah.
Yes, I understand.
So, yeah.
Well, so we would actually need to make a list of issues before October starts, I guess. I think there must be some known bugs or some corrections which would require some minor additions or something.
Yeah, I think that, so you would want to make a list of things. And I mean, there are always things that we find in there, but to have them intentionally made as issues before then. And so it has to be something that isn't a huge undertaking, maybe a bug fix is a good example, where they can come in and do it within a day or less, and then make a pull request, and then we'd review it, and then they'd get accepted, and that would be done, and they'd have their name in the. So last year we got probably about 15 to 20 people coming through and doing stuff, and some of that was people kind of interested in GSOC already, so they were, but that's good because it's it shows that people are kind of using the platform and doing this, engaging year round. So
we could actually take part in GSOD also. I think there is something for documentation as well. So I think our DevoLearn maybe, I mean, it's good, the documentation is good, the existing, everything is good, but we can improve it, obviously. So if we are, it's just a suggestion, I'm not, I don't like documentation personally because I don't like, writing so much thing. But yeah, it's just maybe something we can look upon coming year or coming season of documentation. Yeah.
Yeah. Mainak, you were going to say something?
Yeah, he was.
Yeah, Akshay is right actually. We could use some help in terms of the documentation of the software. So yeah, he's correct.
Yeah.
Yeah, I think I've applied actually to GSOD. Well, we actually OpenWorm hosted a GSOD person in 2019, and he created a continuous integration thing, and which is something that I don't know if we use that much, but that wasn't a DevoWorm oriented. I've applied for DevoWorm for the last couple years, but the way they run the program, they keep changing it, so it's kind of hard to get an actual person to do that. So it's but I agree that I think bringing people in terms of documentation is good, because it can always be improved. We might try to go the route of Hacktoberfest for that, if there are people who and I mean, this would be not just anyone, because not everyone's interested in documentation, but to try to advertise specifically for those issues in places where people might be. But yeah. So and then and we'll keep trying with GSOC or GSOD. Yeah. And so.
Yeah. I mean, if we can attract enough contributors during Hacktoberfest, they will stay throughout the GSOC period also. I mean, till the GSOC period starts, or when the proposal results come. Around that time they'll be, because like this is the trend I'm seeing in my previous organizations also. I mean, I'm still in touch with them, but for example, if you take KD, people would be active till the proposal period. Then from the Hacktober till the proposal period, then they suddenly just go. Then the coming months is just the project is absolutely dead. So we could actually use some help coming months.
Yeah, it'd be good. Yeah, so that Hacktoberfest is one thing that's that's kind of a perpetual thing, but I'll leave it in this area here. Where did I have it? Oh, actually I can create a new one here. That is maybe I'll put that in as an issue. That way we have it to review. So then that's good. And then so we have a couple of things here in the action items. It's game theory for developmental processes preprint, which you saw the talk, and now we want to have a preprint. There's also this paper. Where did I put it? We have the DevoLearn paper that also needs to be written up, or we have the sort of the preprint, but it's not in the state that we'd like to have it to release it. This one here, DevoLearn preprint. So let's put that in action items. So we have these two things here that are preprints that need to be worked on. Follow up on the DevoWorm bibliography. That's something we haven't talked about in a while, but that's all the papers that we talk about, to have them in one collection of references. Update the DevoWorm Weebly site. So I've actually been working on that last couple of days, actually, just kind of like updating some of the content. And again, if people if they want to be featured in the collaborators tab, and you want your picture up, send me a picture in Slack, current picture, and I can post it. The revised ANNs, BNNs paper, this is something that myself, Krishna, and Jesse worked on, and it's something that we submitted to the A-Life conference, and it didn't get accepted, but it got some somewhat semi-favorable reviews, so that's something we can maybe submit somewhere else or revise it. Again, it's on the archive as a preprint right now, but I think there's life in that. Early life and the origins of development theme, it's kind of being developed. I don't know what we'll do with this, if we'll do like a preprint or some other thing. Updates on axolotl data and analysis. This is linked to the thing that Akshay is doing. Next steps for embryo networks and multi-cell systems. This is again this stuff that was done at Works 2021. That's next steps for that, which I don't really have right now. So follow up on origins of embryos. This is not really for, well, it is, I guess. We have some work that we've done on the origins of the embryo, and this is kind of linked to this issue 106. Axolotl montaging, which is related to this 62, and then the stuff that Akshay's doing. Mathematics of DevoWorm, which I've added to here, where we have like these set of models, these set of sort of statements, and now they need to be turned into some sort of paper or something we can present to people. So these are various approaches we use in our.
Could axolotl also be included in this, or?
Yeah, I mean we could. I mean it's really based on methods. So if we had like for the sphere that you're working on, we could talk about the method for mapping those images to a sphere. So it'd be not like the axolotl itself, but
Okay,
sort of
yeah.
the method.
So the math behind it, that's
Yeah.
what you're talking.
Yeah, so it might be like a projection.
I can give you the latex, the math version of that if you want.
Yeah.
I actually have it typed somewhere. I would have to find it, but yeah, I can give you. Yeah.
Yeah, no, that would be great. Yeah, thank you.
Yeah. Okay.
All right, and then let's see. So that's that, and then of course on hold we have a number of things here. I'm not going to go through all of them, but we have things like tutorials and getting data and revisiting some things we talk about in the meetings. And then this create embryo model in Blender. So I just showed you the stuff on the 3D simulations here. And so this is like a idealized version, but one of the things we were talking about, a smaller piece of that, would be creating something in Blender or another platform like Unity, where you
Is that website interactive? Like you can move around?
Oh no, this is a company that does this sort of animation, and I was using this to show kind of like what I was thinking for, like
okay.
Yeah.
I thought the whole thing was interactive. So
No.
Yeah, got you. Okay.
Yeah.
Right.
So this is so this is something that's like kind of been a long-standing thing. So we want to put like create an embryo model and put it into a Blender file, but we also maybe want to do things like visualizing the embryogenesis, taking some of the data we've had as cell tracking data and put it into some sort of context, things like that. So we're kind of still thinking about that. We've had people kind of do things with it before, but nothing really like going all the way with it. So I think steps in that direction would be like the axolotl sphere, that sort of atlas, some sort of embryo model where you have, in a 3D model where you have the cells as 3D objects, and then, visualization like what I just showed with the Animus 3D system there, or their animations. So, and all those things you could do on a number of levels. So that's something else. Then we have these to-do things. So these are things that kind of come up during the meetings. We have the Boring Billions paper, abstract, book topic. We have this Kindle book. We have the droplet shapes, which is, there's an issue over here that's very similar. I don't want to repeat issues, but I think these are a little bit different. But anyways, so we have all these different. This I think is the droplet shapes in broader context. So again, this is on our DevoWorm GitHub repository and group meetings. Go here if you want to look at some of the issues. If you want to add issues or claim issues, you can do that here. So it looks like Mainak left. So okay, thank you for attending, Mainak. So I wanted to talk about this Bacillaria paper quickly. This is a paper that I've been working on for a long time, and this is the psychophysical world of the motile diatom Bacillaria paradoxa. And so I was showing the DevoWorm AI thing earlier, and I talked about this digital Bacillaria project. So this project is something that was done like 2 years ago now. We have a paper on it in our paper collection of our things that we've published. And it is where we apply deep learning and different types of biomechanical analysis to the Bacillaria, which is this diatom. So diatoms are these microorganisms. In this case, it's a colony of single rod-shaped cells. And they have this behavior as a colony where they're doing this sort of stuff where they're moving like an accordion, or they're stretching themselves out, contracting. So it's a very interesting organism in terms of movement. And what we were doing, we were identifying cells and doing cell segmentation, identifying the trajectory of different cells moving against one another and all that. This paper follows up on that. It's more speculative, and it talks about sort of what the potential psychophysics of this diatom are. So what do you mean by psychophysics? And what I mean is that there's no brain in Bacillaria, but it does have these properties of sort of what we call non-neuronal cognition. And so we talk about non-neuronal cognition in this paper. We talk about the different aspects of, it's a metaphor, but it's also a way to explain some of the mechanical properties of the colony and how it's moving. And critically, there are different types of measures you can use and different types of techniques that we use in measuring perception in nervous systems that can help us understand this system. It doesn't have a nervous system. The system has this sort of network of filaments and other types of components that allow it to move collectively. And so the question is, how does it do that? Can we model it using things that are, from psychophysics or from like cognitive science and neuroscience more generally? So we go through a lot of these like different aspects of the neural architecture, different other organisms with a similar type of interpretation. So people have looked at slime molds, Physarum, in terms of non-neuronal cognition. They've observed different types of learning, like habituation. They've observed this sort of thing, non-neuronal cognition, simple chemical systems, such as protocells. They've observed dissociative learning and habituation in different types of minimal cognition systems. So there are all sorts of different examples. A paramecium as well, where they can regulate their swimming movements. So there are all these like systems that don't have a brain, but also have this type of behavior. So we kind of go through some of the methods that you might use. And again, this isn't really a data-driven paper. This is where you just talk about a lot of these different things and bring them to this audience. So we're borrowing from the neuroscience literature and the cognitive science literature. We're looking at things like predictive processing and Hebbian learning, and also, different measurement techniques like using the Weber-Fechner law, which is a law of light stimulus intensity. So this is something that we can actually link directly to the diatoms' behavior because they too respond to light intensity. And so we can propose a law that's relevant to that.
Is this a paper, by the way? I'm just curious. Like, you are going to submit this to some conference, or?
Yeah. So this is a draft paper, and I don't care about being secretive about it. This is something we're working on, and it's not really all done. It's not done yet, but it's being submitted. It'll be coming out as a preprint in the near future as soon as it's finished. Yeah. So I wanted to go over this to give people in the group.
Is there some practical examples, visualizations, which we could show, like, that supports these equations? Or?
Yeah, so I have some diagrams later, but I don't have anything that's really... This is a thing I think that's missing, is that it needs some sort of like... We didn't really... See, we were going to put a data analysis in here, but we couldn't quite get what we wanted. So we just went in this direction, but I think it's missing like some sort of simulation or some sort of like making this more practical.
Yeah.
And I don't know how to do that necessarily. And that's why I wanted to bring it up to people, because I wanted to see if people had suggestions on this.
Would there be data? I could actually volunteer to help you on this, but I'm not sure of the biology. But if there is something that needs to be, simulations or something which needs to be shown via these equations, which I can plot or learn through behavior of these diatoms, I can actually help you. So
okay.
So if you have, actually free.
Well, there are some things I don't have anything right now, but it's something that we can talk about offline, because it's like something I have to think about what would be the best way
Of course.
Yeah.
Sure, Bradley.
Yeah. So, and then this goes through, different methods, and then we come up with this thing that, this is something that's novel about our approach, is we've come up with this thing called a collective pattern generator. So this is a Bacillaria colony, and it's being exposed to light, and they get exposed sort of at the ends of the rod here, but you have these points that are like intercellular connections between the cells, and so these are things that we're sort of modeling with a connectionist network. So as it moves, the network the weights of the network change here in between these nodes. And the idea is that, the intensity it changes, but also with like the movement of the colony it changes. And so then, kind of get into a bunch of different methods here of looking at different behaviors like halting and dynamical states. And these are some some simulations. They're not like dynamical, but they kind of show some of the aspects of like what a halting pattern would look like versus like an oscillatory pattern, and showing this in different cells, and then showing the addition of noise, and then looking at this as sort of an attractor point, and then kind of going through like the relevance of phase transitions to movement, and then I think that's about it. There's some discussion at the end about the broader picture in terms of, is this really intelligence or is this pseudo-intelligence? Because, it doesn't have a brain, and we're saying that it has these properties of cognition or aneural cognition. So, how do we know whether it's truly intelligent or pseudo-intelligent? And there's this discussion here on this. And then that's it. And there's some quite a few references here. Probably we'll add more, but that's what we have right now. So just, this is for people, anyone in the group, if you want to contribute to this, let me know. And Akshay, I'll get back with you on like some of the more detailed points of this.
Of course, yeah. Via Slack, yeah?
What was that?
You will be contacting via Slack?
Oh, yeah, Slack. Yeah.
Yeah, sure. I actually, had to change my distro, and it's all gone, so I have to reinstall stuff again.
Yeah.
Yeah, sure. I'll do that.
Okay. So, all right, now I think we'll move on to papers for the week, and I'm going to start with some new embryo images that I found online. So there are a couple of interesting ones here. I think this will give you maybe a little bit better idea of some of the organisms we talk about in the meetings. So the first one is an axolotl, and this is in what they call the Nonneuronal stage. So this is a pretty early point in axolotl embryo development. This is where it's sort of forming its neural tube. So it's forming the tube that's going to form the brain and the spinal cord here. So it's folding over. And so these are the Nonneuronal stages. And this is the person who took this picture, Dr. Crystal Rogers. And this is it's an axolotl embryo. This is a close-up here in their experimental apparatus. And then there's this other one, which is a 6-day-old zebrafish larvae. And this is from the Zebrafish Rock Twitter account. This is Oscar Ruiz, who's this is the person who took this image. So this is a close-up. This is a 6-day-old zebrafish larva, the developing face. So this is what it looks like at 6 days of a zebrafish larva in the face as it's developing. So these are the cells. They're stained, and you can see the staining, how it's different here, and you have this face coming up. This is actually fairly far along in zebrafish development, but you're starting to get the features of a face here. So this is, and then finally you have this tree frog embryo, which is a really nice image. And this is this comes from a paper, this paper here, which talks about multimodal mechanosensing, and it enables tree frog embryos to escape egg predators. So this is where they're looking at this embryo and how it's able to escape a predator. So they actually, after they hatch, they form these tadpoles. So they don't form the full amphibian yet. They have this tadpole stage. So post-hatching tadpoles use something called lateral line neuromasts, which are these cells along the sides of their body, and it forms a line down the body. And these cells detect water motion. So we have our lateral lines. In fishes, there's a lateral line system which detects anomalies in the water flow around it. So that's how they can navigate and detect prey. And tadpoles have the same system. We ablated neuromass function with this gentamicin, which is a toxic substance, to assess the role in hatching responses to disturbance. And so prior to vestibular function, this neurally eliminated the hatching response to a complex simulated attack cue, egg jiggling, revealing that neuromass mediated early MCH. So this is where they're actually introducing this predation, which is where some predator tries to break into the egg, and the tadpoles can detect this, and they're able to respond to it. So that's what they're talking about in here. Now, the key to this paper, the reason I pull it up, is because you have these nice images of the tadpole embryo. This is the front here, stage 29 at 5.63 days. This is stage 30. And then there's some other, this is the hatching response here. But then there's some other images here where they actually trace out some of the tissues and some of the anatomical features. So these are the nares, which will become the nose, the supraorbital area, which is above the eye, and you can see these things forming here. So you can see that. All right, so that's the new embryo images. Let's see, what else do I want to talk about here? So this is something from a developmental biologist who's not really impressed with genomics as it stands today. So making this comment, this notion that genomes build cells, tissues, bodies. I understand that by repeating this mantra, we get to believe it without questions, but it jars us with the reality of a developing embryo. Cells use genes and are not directed by genes. Shape and structure are not in the genes. So this is something he advocates for, the role of, the cell at the cellular level, at the organism level. And that's something we talk about a lot in this group. And so the idea is that the genome isn't really the set of instructions per se, or a blueprint. It's more like things that are sort of encoded at the genomic level, something that is, ensures that things kind of are built. It's not like, don't think of it as, I wouldn't even think of it as a blueprint, but it's a way to ensure that things always develop in a similar way. But they can be expressed in many different ways. And so this is the kind of the point, that there are a lot of things, not only in terms of gene regulation, but things at the cellular level and the organismal level that contribute to an organism's what they look like. And so this paper talks a little bit about brain evolution by brain pathway duplication. And this is a nice paper because it kind of talks about some of the mechanisms for brain pathways. And so this links the genome and some of these other processes. So understanding the mechanisms of evolution of brain pathways for complex behaviors is still in its infancy. Making further advances requires a deeper understanding of brain homologies, novelties, and analogies. It also requires an understanding of how adaptive genetic modifications lead to restructuring of the brain. So they found that recent advances in genomic and molecular biology have provided exciting insights into how complex behaviors are shaped by selection of novel brain pathways and functions. So they review these different things, and they kind of come to this new hypothesis on one mechanism that may contribute to nervous system evolution, and that is brain pathway duplication. So you start off with a brain pathway in development, and those brain pathways in organisms that evolve, they don't just generate new pathways, they tend to take pathways and duplicate them. So this is something that happens in the genes. So in genetic evolution, you get like gene duplications or other things, or you get things expressed twice in the same brain. And so this is one way that evolution uses to produce more complexity in the brain. So like gene duplication, we propose that whole brain pathways can duplicate, and the duplicated pathway diverges to take on new functions. So that means that you have two pathways that have the same common origin, but that those two pathways then will diverge in terms of their function. We suggest that one mechanism of brain pathway duplication could be through gene duplication, although other mechanisms are possible. So they talk about their interest is in vocal learning and spoken language and song learning birds and humans. So they're looking at the differences between these two pathways, or sets of pathways in birds and humans. And so birds and humans actually have this sort of common function of generating speech and song, which are very similar in terms of forming grammars and things like that. So, but they have totally different brain pathways for this. Humans have a bunch of speech areas that are connected, and birds have a bunch of song areas that are connected. And they have actually looked at this group of people have looked at this problem, and they've seen, these pathways don't look anything alike, but they've actually been able to dissect sort of the common ancestry of these. And so they're able to make these homologous comparisons between what's going on in birds and humans. So explaining these complex pathways, so this is a pathway in humans, this is the language pathway, this is the song pathway in birds, and you can see that they don't look anything alike. On the other hand, what you have here is you have a duplication of pathways. So you get pathways that start off from a common ancestry, and it gets duplicated, and then the function is modified so that you end up with wildly different phenotypes. You get circuits that converge. So these are the two different types of brain pathways here. These set of brain pathways are homologous, but they have different structures, and they form by duplicating themselves and then differentiating. So this is an example here, more pictures of the evolution of the song system in parrots owing to sequential pathway duplications, the ancestral motor pathway, which is this light green area, with the posterior motor connections in black arrows, and the anterior motor connections in white arrows. And so this shows how this whole entire circuit sort of comes together in evolution. This parrot core song system proposed to have evolved out of the pre-existing motor pathways through duplication. The parrot shell song system, yellow, which are these areas, proposed to have evolved out of a partial duplication of the core song system. So these different areas that have different functions kind of evolve out of a core, and then they kind of expand the phenotype expands to multiple similar pathways, and then it forms a larger system that's capable of different types of specialized behaviors. So that's that paper. And I just wanted to go through that just to give people an idea of what what this kind of what this looks like. And so then I'm going to jump to another paper on amoeba-like creatures form digestive fronts to consume their prey. So now we're moving back away from brains, and we're moving towards these kind of collective behaviors. And so in this case, we're looking at the organism Placozoa, which is the simplest multi-celled animal ever described, that lives without muscles, nervous tissues, or digestive systems. This is from Science News. To eat, these creeping bundles of cells spit out enzymes that externally digest algae floating in the water, which they then absorb through their transparent bodies. New research finds that when Placozoa eat, they do so in groups of as many as 105 individuals, forming moving fronts to collectively devour the algae in their path. So little was known about the behavior of these flat sand grain-sized animals, even though they live in oceans all over the world. So to learn more, the researchers set up an aquarium of this, and once the placozoa were released into the tank, the researchers recorded their movements for a week. And so these amoeba-like creatures began to eat the algae. They congregated in groups of varying sizes. This is based on a paper in Frontiers in Ecology and Evolution. Communal feasts might help placozoa digest their food faster than they would in a solo case. So they form these groups, they feed on a common resource, and they form these fronts that then consume the food. They cooperate, basically, to consume the food, and it helps the entire group of placozoa. But they point out that social eating does not necessarily mean that creatures are cooperative. Freeloaders could also join groups and absorb nutrients without contributing to their own digestive systems, or digestive enzymes. So this is where, all of the organisms are contributing digestive enzymes to break down these algae and eat them. And there could be freeloaders in the group who don't contribute any sort of enzyme, but then consume along with the group. So this is something we could actually model using game theory. It's a common problem in game theory are freeloaders, and especially if you're looking at cooperation and the evolution of cooperation as well. Nevertheless, the discovery that these simple organisms eat together suggests even the earliest animals may have had social tendencies. So that's an interest. That's not the paper itself. This is the Science News article. So if you're interested in the paper, I can pull the paper up. And then this last paper I'll talk about is this dislocation as a developmental mechanism in Dictyostelium invertebrates. So this is an interesting paper. This kind of talks about Dictyostelium, and it's a sort of a mold. So it's not, it's not a very, I guess it's a complex organism. It exhibits a sort of collective behavior. And, this is then, of course, they're saying that this location works in this, it's apparent in Dictyostelium, but also vertebrates. So we're talking about, again, widely divergent phylogenetic parts of the tree of life. So the abstract of this paper reads, the excitable cells of Dictyostelium discoideum shows traveling waves as signaling and generate a variety of complex waveforms during their morphogenesis. Important among these waveforms is the 3D spiral or scroll wave, which has been proposed previously to have a twisted variant, the turbine wave. So this is where we talk about these different forms of and you have one of these collective organisms. It's a collection of cells, and their morphogenesis exhibits these type of waves that go through and coordinate things. So these are excitable cells that are connected together, and the excitable cells sort of get excited in sequence, and they form these waves across the morphology. So important among these waveforms is the 3D spiral or scroll wave, which is something that's a physics, it's a well-known physics wave. They've classified different types of waves in excitable medium. And then there's this turbine wave, which is a variant of it. Herein we argue that this organism's scroll or concentric wave territory containing presspore and prestalk cell types, so these are two different types of cell in this organism, can undergo dislocation, a wave field that initially controls aggregation of a whole developing population. And then these cells split into two. So it's this idea that you have this population, there's this wave that goes through the population, and then it splits the population into two parts. This process leads to discontinuity between two connected domains of wave propagation and a specific phenomena, including high-frequency concentric pacemaker activity by the slime mold scroll wave tip. So this is a slime mold, and it has these cells, and it's there are all these different types of patterns that are splitting it into different parts. And then they're actually still connected, but they're split, and there's this discontinuity. So you get this, you get almost like these little modules within the colony And then these high-frequency concentric pacemaker cells, which keep a rhythm, they have a regular rhythm. They're part of this excitable cell activity. The resulting morphogenetic events reveal a unique mechanism in morphogenesis. So when I say excitable cells, I mean like neurons. Neurons are like muscle cells, like heart muscle, things like that. Those are all excitable cells. And they have this, ability to generate pulses of electricity. And you can have, like, a lot of excitable cells are coupled like in brains, but you also have this in slime molds where they can share this sort of excitability. And so that's what they're talking about in this paper. And so they're pioneering observations by John Bonner and other people. Brian Schaefer in the 60s and 70s demonstrated that the chemotaxis to CAMP and CAMP signal relay, which is this chemotaxis, are where cells respond to these chemical signals and move accordingly. And so we've talked, I think, about chemotaxis a little bit in this group, where they have a chemical signal and there are these waves that are controlled by these molecular components, and the chemotaxis follows this, so it sort of coordinates the movement of the cells. So this mediates aggregation, and so they kind of go through this whole model here of what they're talking about. Some people propose that this, what they call a slug, which is the actual collective, is organized by a twisted scroll wave. So these waves actually are well characterized, and they also serve to organize the entire colony, or what they call slug. And so I don't know how they make the connection to vertebrates, but this is a picture of the scroll waves and other types of waves that they talk about here. So this is a type of reaction we've talked about a little bit in the group, called the Belousov-Jabotinsky reaction, which is where you can do this with a chemical medium, where you have some chemicals and you can create a reaction that generates these patterns here, these wave patterns. And they're concentric wave patterns that come out from a source. So you end up with this sort of reaction where there's all this pattern formation, but it isn't life that's doing this, it's just a chemical reaction. So you see these waves. So they talk about waveforms here, where they illustrate the waveforms in a simple excitable media. So in the chemical case, it's an excitable medium. When you have cells, you can also have these excitable media that are organized in this wave. So here in this example here, I think in F, this is where they propose what the scroll waves look like in slugs. So you have this slug is organized in this way where there's this scrolling. So some of these scroll waves are bent and can also have their ends joined in torus rings. So this is the dislocation aspect of it. So observations that argue against trusted scroll waves for dislocation. So scroll waves and dislocation are competing hypotheses during tip formation. So this is an example here where they show the concentric centers. These are relevant sections of expanded spherical waves. So this is here. It's an example from A where they have these concentric centers. And then, so this is A is the concentric ring wave of relay initiated by a point source pacemaker. So this is a pacemaker cell that's generating this pattern. And then B is where you have other cells that are controlling this spiral wave. And so you can see this is a little bit different in B. All right. And so I'm trying to figure out where they talk about vertebrates. I think that might be at the end. Okay, here we go. This simple morphogenetic system may yet provide general inspiration indicating general principles for embryonic development. So this is where they're expanding it out to embryogenesis. Recent findings strongly indicate that chemotactic movements organize the vertebrate gastrulation, and that these timed gastrulation movements are the key to making the vertebrate body plan. So they're making a link between this scroll wave activity and gastrulation. The situation regarding oscillatory and relayed signaling is more complex but interesting. There is now much evidence that waves, coupled oscillators, and relaying are important in the development of metazoan animals. So this is something that we've talked about with respect to differentiation waves in development. An early model proposed a progressive phase shift between two traveling waves originating at the organizer as a mechanism providing positional information in the vertebrate gastrula. So this is where you have these waves that are organizing things and they're providing positional information. So we've talked about positional information as well, and this basically gives some sort of spatial, making things spatially explicit in the embryo as it starts to matter. And so another early study actually detected synchronized oscillatory morphogenetic movement in the chicken gastrula, and another study detected scaling controlled by a regulative phase gradient as predicted in the Goodwin-Cohen model. So there's a sort of connection between vertebrate embryogenesis and what's going on in the dictyostelium. So that's all I have to say for today on papers. Okay, so Akshay has to go. Well, thank you for attending, Akshay. And so I guess that's our meeting. Thank you for attending, Akshay and Mainak. And I'll be back. We actually won't be meeting next week. I'll be taking the week off, but then the week after we'll be talking about, we'll be trying to move forward on some of these issues, preparing for Hacktoberfest, talking about more papers, and maybe people will present things as they have time.